হ্যাঁ, ইউটিউবে একটা কাউন্টডাউন মতন দেখাচ্ছে, 5 মিনিট 26 সেকেন্ড, 5 মিনিট 25 সেকেন্ড এরকম করে।
এখন দেখো।
হ্যাঁ মানে, হ্যাঁ ওইটাই দেখাচ্ছে, মানে 5 মিনিটের
না
মধ্যে শুরু
আমাকে তো
হবে সেটা।
আমাকে তো ইউটিউবে স্টার্ট করে দিয়েছে, মানে অনুপমদার এইটা দেখাচ্ছে।
হ্যাঁ, ফ্লায়ারটা
হ্যাঁ,
দেখাচ্ছে।
ফ্ল্যাটটা দেখাচ্ছে,
আরেকবার
আর
ওটা বন্ধ করে খোলো।
মেসেজও আসছে, একজন মেসেজও
বন্ধ
করেছে।
করে বন্ধ করে খোলো। আমি তো ইউটিউবে অনুপমদা যে এটা সরাচ্ছে ওটা দেখতে পাচ্ছি।
না, এখানে কিন্তু দেখতে পাচ্ছি না সেটা।
দীপশিকা দিয়ে রিফ্রেশ করো, তাহলে এসে যাবে। ওটা বন্ধ করে আরেকবার চালাও।
হ্যাঁ, রিফ্রেশই করছি। হ্যাঁ, রিফ্রেশ করছি, আমারটা এখনো আসেনি।
Yes, Arpan.
হ্যাঁ,
It's my slide is displaying right now. I think looks like opened here.
হ্যাঁ,
Yeah, sounds good actually.
both in same, মানে in YouTube and also this both same.
Yes.
Okay, thank you. Hope we shall
Yeah.
success.
Yeah, hopefully should go. I think this time should go very smoothly. Your flat looks nice.
হ্যাঁ হুম দিনকে ফোনে পাচ্ছ না? Anupamda, we will just wait for 1 minute more, then we shall start again.
Sure.
Just 1 minute. Prabhuto, please start.
A very good afternoon to everyone who is attending this webinar. Just a gentle reminder, we were actually supposed to have this webinar on 17th, but due to some technical problems, unfortunately, we could not proceed on that day. So here we are back again with the webinar today. Without taking much time, let me extend a very, very warm welcome to all our participants and our respected speaker and all the dignitaries who are present with us today. Before we get on with our session, may I kindly request Professor Sanjeev Ghosh, Professor of Eminence, Advisor to Vice Chancellor, Adams University, to kindly address our audience for today. Sir.
Good afternoon to everybody. Am I audible?
Yes, you
Yes,
are.
sir.
Okay.
Yes, sir. Yes, sir.
Good afternoon to everybody, to all students of Adams University, faculty members of Adams University, as well as all the other participants, and obviously Dr. Anupam Bandyopadhyay. Thank you very much for agreeing to this particular webinar. And after COVID-19 pandemic, during this pandemic period, the Department of Chemistry and also the School of Science are arranging lots of webinar for our faculty members as well as for the students. And also we are inviting participants from outside. And for example, the last seminar was by Professor John Mary Lane, who is a Nobel laureate. The last Friday we had this. And so today's topic is very important. As you see in your screen, the technology-assisted antibody mimetic discovery. This COVID-19 has opened up a lot of research, particularly in the field of many people are trying to, get a vaccine developed. Many are trying for particular drug which will be suitable, proper drug, and then also many are trying like this one, antibody mimetic things, and also plasma therapy and all kinds of things. People are trying all over the world, and we are very fortunate that Anupam has agreed to talk something about this technology-assisted antibody mimetic discovery. Hope everyone participating in this seminar, students, faculty members, and the outsiders will be benefited much from this and could develop on their own something for this particular COVID-19 research. With these little things, I think I hand over to Prabhuddha for further continuation.
Thank you, sir.
Thank you.
Yeah. We have now amongst us Dean, School of Science, Professor Bimal Kumar Sarkar. May I now request Professor Bimal Kumar Sarkar, sir, to kindly address the audience. Sir. Can we have Professor Sarkar address the audience?
Hello, sir. Hello, sir.
Yes.
Yeah. Yeah. Sir, now you're having Professor Sarkar with us. May I request Professor Sarkar to kindly address the audience? Sir, can you kindly say a few words?
Some problem is 1 minute, just 1 minute. Just 1 minute.
Okay.
Yes.
Am I audible now?
Yes, sir. You are
Yes, sir.
audible.
Yes, sir.
Yes.
Audible.
Sorry, just really, it's pleasure for us that we are now just in a continuation. We are just doing webinar today, and post COVID-19, what will be the just opportunities and challenges? Just it will be discussed today, and I think this is a right moment also, and I wish a very big success and very conclusive, and at the end very conclusive moment will reach there, and I wish all the success of today's event. Now over to Prabhuddha. Thank you very much.
Thank you, sir. Okay. So before we go on to our today's talk, let me just in a very brief introduce our today's speaker to the audience. Today, we are very fortunate and honored to have amongst us Professor Dr. Anupam Bandyopadhyay. Now, just a short CV that I'd like to introduce. Professor Bandyopadhyay received his PhD degree from IISER Pune under the supervision of Professor H.N. Gopi. His works were mainly on polymer chemistry and bioorganic chemistry. He then joined Boston College as postdoctoral fellow in 2013 with Professor Jianmin Gao, where he worked primarily on selective membrane lipid recognition. After that, he joined MIT for further postdoctoral experience in Professor Badley Penteluke's lab, working on development of technology on high true output screening utilizing synthetic proteins. Now, along with that, Professor Bandyopadhyay is also the recipient of various prestigious awards and recognitions. To name a few, he has received the prestigious NIH-aided Boston College Postdoc Research Fellowship in 2013, Young Investigator Award at the Peptide Gordon Conference CA USA in 2016, STAR Postdoctoral Fellowship Novo Nordisk in 2016, and Postdoc Bridging Fellowship awarded by NCBS TIFR in 2018. He has been also awarded with the Ramanujan Fellowship, which was awarded by DST SAR. He has published quite a few papers in various reputed international journals with high impact factors like PNAS, JACS, and he has been working as assistant professor at IIT Ropar from January 2019 onwards. With this brief introduction to our speaker, may I kindly request our today's speaker, Professor Bandyopadhyay, to start his presentation, which is entitled as Technology-Assisted Antibody Mimetic Discovery, Prospect in Identifying SARS CoV-2 hits. Sir, it's the stage is for you. Thank you.
Thank you, Prabodh, for the nice introduction. And I would like to first thank the organizing committee of Adamas University, especially to Dr. Arpan Manna. And this is my pleasure to give a talk which actually my research group are pursuing kind of a technology, which is, I think it can be a powerful for discovering antibody mimetic or any kind of epitopes or peptide therapeutics or diagnostics. So today, what I'll be talking about, the technology-assisted antibody mimetic discoveries. So the technology, what I'll be talking about, which is high throughput technology, screening technology. So I will basically talk a little slower in speed than usual. And the first, fundamentally, I'll talk about the what is antibody mimetic, and then talk about the technology, how you can do it on bench, the screening technology. Further, I'll switch the gear to the prospect in identifying SARS CoV-2 hits by using this technology. So let's begin with that. So first you need to know what is protein. So let me go back. So protein is nothing but a heterobiopolymer, so which actually folds in a certain structure which can give a particular biological function. As an example, you can see, you are definitely very much familiar with this particular picture. It's an omelette, egg omelette. The white portion is completely comprised of the protein. The 10 major proteins are very well known. It is completely comprised of, if you do not consider the salts as well as the water. So the major contribution of this particular protein is, I'm showing here as a ova-albumin. So you can see I'm showing here is a kind of a cut structure. It's a huge chemical formula. So that's why people, for the convenience purposes, it's made a cartoon. You can see there are some helix structure, there are some beta sheets, and also the turns, which actually gives the structure. Something is going on. Someone is writing in my. Okay, anyways, I'll continue. So this is another example I'm showing here. The lysozyme, it exists into this particular white area, white. So this is the lysozyme, one of the protein known as an antimicrobial protein. But what is the common between them? As I said, they have a structural a diversity as well as the functions are different. The commonality is, if you chop out a small portion of a protein, you will see this kind of chemical structure, where you can see that the amino acids, they join together and form this kind of particular protein. And the common feature is they have the imide bond. The 20 natural amino acids are very well known. These 20 amino acids are actually joined through the imide bond. And amino acids are the building blocks. It's very well known for the proteins and peptides. Then how do you distinguish between peptides and proteins? As I said, protein is basically having a tertiary structures, which is a folded structure and having a biological function. But peptides are not necessarily has to have a tertiary structure. It can be a random coil or it can have some secondary structure. But typically or arbitrarily, people call if the amino acids counts are less than 50, then we call them as a peptide. But beyond that, if it's a very lengthier chain, then we can call them as a protein. But it has to have, again, keep in your mind that it has to have a proper tertiary fold as well as a biological function. Now we will go to what is antibody and its function. So antibody is nothing but, again, a protein. It's a very gigantic protein. As I am showing the structure, it has basically a heavy chain as well as a light chain. If you look at the structure, how it looks like, this is showing here in the zoom. And the another structure I'm showing here, this portion is specifically called a hinge region, that it is much more flexible actually. And the most important part, what I will be talking about, which is this particular region. This is known as the FV region, the Fab region of the antibody, that is actually responsible to bind to the antigen. So antigen means they are pathogen. So antibody are mainly produced in our plasma by cells, and these are our immune system that fights with the antigen using this particular protein molecule. Now I will talk about how we fight against virus. So let's say consider this yellow color, these balls are the viruses, and it actually displays the antigen, different kind of protein, and the antibody comes and bind with this particular antigen. And you can see it as it has the two hands, as I showed it has two hands, it can form this kind of polymerase structure, like it's assembly just, and it's called a neutralization process because it makes a huge cluster kind of. So that's why it neutralizes the virus, that is the way. Now next, I'm coming to what is antibody mimetic. So antibody mimetics are organic compounds, like antibodies can specifically bind to antigens, but that are not structurally related to the antibodies. That means you can see the antibody, what I showed, it's a very gigantic structure, but antibody mimetics are kind of their function or specificity bind to antigen that is similar, but not structurally related to the antibodies. That means their molar mass is near about 3 to 20 kilodalton. This is much smaller than antibodies are typically 150 kilodalton in size. So it's almost like a 10 times lower in size. So these are the kind of, I'm showing some structures that are known as antibody mimetics. So apibodies, nanobodies, apimers, affilines, monobodies, avimers, DARPins. These are kind of like given the name with the different small structures which are known as the antibody mimetic. So antibody mimetics can be nucleic acids, also small molecules are sometimes can be considered as antibody mimetics, which can actually make the function of antibody not related to the structure. So now we are moving to a special type of antibody mimetics, which is known as a paratope mimetic. So as I already discussed, the antibody looks like a Y-shaped big protein, and this is the portion is known as the antibody Fab region and mostly responsible for binding with the antigen. And these actually also people call as a epitope. That particularly I'm showing here as this particular the loop kind of structure, which is in a purple color, red color, or orange color. These are the particular region actually binds to the antigen. Now you consider that why do we need then? These are the huge portion. We don't need that. If we can mimic that particular structure, which is nothing but a peptide sequence, that can be able to mimic the antibody function. So peptide chemistry along with the organic synthesis play a very crucial role if you want to mimic this particular structure. So therefore, I just called that peptides are astounding gift from the nature. Peptide has been taken in a new direction for the drug discovery in the pharmaceuticals companies. And peptides has been used for the therapeutics purposes, theranostics purposes, molecular carrier. People know about the drug delivery systems, as well as nowadays peptide vaccine are very popular. So you can take a look. I'm showing here a peptide therapeutic markets. This is the US market is a keep increasing the demand every year. As you can see that 2025, it's going a really huge demand, continuously growing from the, if you start from the 2014. So blue color is basically the generic peptides, and the dark navy blue color, this is representing the new innovative peptide that the people have been discovering. And this is the pie map. As you can see, the distribution, particular what purposes it has been used, peptide therapeutics, that you can see mostly the cancer is the major target. So other than that, many other things people have been using, as you can look at through this particular citation. Now, what is the challenge with this peptide? The challenge is the oral availability as well as the proteolytic stability. So we have a lot of enzymes that can proteolytically degrade the peptides in this particular bond, this amide bond. And there will be no more identity of any that molecule you make it or peptide therapeutic you make it. So therefore, that is one of the big challenge. But again, the chemist can actually play a crucial role here to modify this peptide such a way so that enzyme cannot recognize. So that has been already considered, and 4 major synthetic modulations people has been considered. And as I'm showing is the peptide has been modified such a way that the chiral center, the center has been changed to the L amino acid or D amino acid, or maybe peptide has been cyclized so that it will be less flexible to recognized by the enzymes. Backbone modifications, as I'm showing here one example that N is methylated. Also the side chain modifications, the natural side amino acid side chains are 20. And now this, if you change in a different thing, so the enzymes may not be able to recognize. That is the way you can basically modulate the peptides so to restrict the proteolytic degradation. So now the question is, how do you develop a high affinity and great selectivity antibody mimetic against a biological target? As I talked about, it has to have a high affinity as well as the selectivity that antibody does. The function what antibody does is great selectivity as well as the high affinity, which is not that easy to generate. So that how could you do that? It's very easy to answer, to understand the nature, but it is not actually that easy in terms of the practical. So I think the factors which are responsible for the specific and tight bindings are mostly if you look at the molecular recognition process, they mostly involve the non-covalent interaction with a proper structural integrity, which can actually cut off the entropic penalty as well as the induced feed. And the induced feed, this word particularly used for the enzyme substrate binding purposes. And these non-covalent interactions are mainly governed by pure ionic interaction, as I'm showing here. This is kind of negatively charged, positively charged, dipole interaction, hydrogen bonds, hydrophobic interactions, as well as the van der Waals interactions. So if we want to know or mimic a particular receptor or antibody mimetic, you need to know what kind of interactions are there through the crystal structure. And you have to also scrutinize the amphitheticity that is basically having either hydrophobic or any kind of ionic interaction, where you need to tune and what direction you need to make an integrity of the structure. That is the way you can actually make a receptor. So how do you develop a high affinity? Again, I'm asking this. There are 2 ways people have been doing it. One is the rational design. So you actually try to understand how molecular recognition is going on, understand the hydrophobic interaction, molecular other kind of van der Waals interaction, ionic interaction, hydrogen bonds and other things. that is the way you can think of that how you can design, or the other strategy could be the de novo high throughput screening. De novo high throughput screening means there is a basically not known any molecule that you know that is going to do that purpose, which is completely a new molecule you are going to discover. So this is kind of a high throughput screening you have to do with a infinite possibility of molecular diversity or chemical diversity. And then you do the screening process. Further, you will end up with some molecule, they are actually doing the purpose, what you're looking for as an antibody mimetic. Well, so now let's look at the thing that this is the one example I'm giving it, how people have rationally designed a particular, this kind of receptor or epitope, you can say. This is one example I'm taking, which is lactadurine protein. It's one of the protein present in milk. And the spike 3, as I'm showing here, the spike 3, this is one of the portion which is here zoom structure. This zoom structure is showing that it binds specifically to the phosphatidyl serine. This is one of the phospholipid, is very well known biomarker for the apoptotic cell. Apoptotic means it's a cell programmed cell death. So when cell dies, basically the particular this phospholipid comes outer surface. And this is the one of the biomarker people can track that cell died or not. So what is the application? Let's say after post-cancer treatment, you want to check that after chemotherapy, the cells are died or not. That could be one of the way you can track it. So people use a very well-known protein. It's a huge protein, is named as annexin 5 with a particular fluorophore or kind of radionuclide. But it is a very big molecule and it's not a cost-effective way you can develop this kind of particular protein. And also it has a lot of other side effects when you treat with them. So rather, if you have a very small peptide, that could be cost-effective as well as it may not induce a lot of toxicity. So let's take a look that how is it. This is basically, I'm showing the lactadurine site where the phosphatidyl serine head group bound. This is the dot structure. And with that, you can take a look that each amino acid, if you basically join and make a cyclic kind of peptide, like the way it has shown here, this is the particular peptide, cyclic peptide can specifically bind to the phospholipid head group. And this has been published in a JACS, as you can see, and it is very selective to this particular protein, but affinity wise, it's not very high as compared to the lactadurine protein. So this is one of the big challenges, as you can see. How do you now, after that, increase the affinity? So now the next I'm coming to the popular high throughput peptide screening technology, which is already known, or the recently people have been discovered. The first one, what I'm showing is one bead, one compound. This I'm going to talk about, this technology, as well as the solution phase screening. That is also recently discovered, that I will be talking about. The spot synthesis, what I'm showing at the right, this is already very old and very well known and very limited peptide library you can make it. That's what it is. Actually, people are not being considering for the de novo discovery purposes. So there is another technology, which is the help of the biotechnology, specifically the phage display. With the help of genetic technology, you can genetic modification, you can actually let the phage express a different kind of peptides or proteins. Now you can screen and then find out which is particular peptide or protein is the binder of your target. But I'm not talking about this biotechnology, this process, as our lab is working on the chemical synthesis as well as the chemical synthesis related technologies, high throughput screening. So we'll talk about the first, this technology, which is by the chemical synthesis, one bead, one compound, as well as the spot synthesis, that what I have shown here. So the one bead, one compound and the solution phase. Now take a look that you need to know first if you want to synthesize this kind of library, you need to know what is solid phase peptide synthesis. The solid phase synthesis is solid phase peptide synthesis, but this is chain assembly by protected amino acid on the solid support, on the resin. So now what I am showing here is the Adamas, IIT Ropar, Chandigarh, Amritsar, Kolkata. All these things, what I'm showing is nothing but you can consider them as a peptide. This is a peptide sequence. So each letter denotes a particular amino acid. A is alanine, D is aspartic acid, A is alanine, M is methionine, A is alanine, S is serine. IIT Ropar, isoleucine, isoleucine, threonine, proline, arginine. Like that way, you can see. So Adamas, IIT Ropar, Chandigarh, Amritsar, these are all in a natural amino acid to non-natural amino acid. So that will allow you to make it a different kind of a non-natural amino acid. So you can give a name. So that will give you a Kolkata is another peptide sequence, which is very different from the Adamas, IIT Ropar, Chandigarh, Amritsar. And Kolkata is a special now. So that can be only done by the organic synthesis or the modification of the natural amino acid to the unnatural amino acids. Now take that how peptides are solid phase synthesis being done. So now, people, yes.
এই যে এইগুলো আসছে না মানে
So why is this coming? I don't know.
হ্যাঁ ওটার উপরে ক্লিক করে ক্লিয়ার করে দাও তো আমার মনে হয় চলে যাবে তুমিই পারবে জাস্ট ক্লিক করে একটা ক্লিয়ার অপশন আছে somebody is doing
yeah I don't know it's really the screen is going bad. So can anyone do that? I'm admitting Jyotirmoyi Panda. Who is that? Jyotirmoyi Panda.
That's not a problem, but please Jyotirmoyi Panda, what he is doing actually?
So asking for to admit or see waiting room. Something has entered the waiting room for this meeting.
That's not problem. Oh, that we will see.
Okay, fine.
Please
so
click on
who is doing that? I don't know. It's making his noise actually.
Just click on it and just you see that anything is coming.
Screen আস্তে আস্তে খারাপ হয়ে যাচ্ছে অর্পণ।
Screen option, just screen option.
Screen
clear আছে দেখো black white
pointer option.
Can anyone do that?
laser pointer eraser আছে একটা there is eraser option.
Eraser is not
maybe
active.
eraser option টা দেখো তো
ওরকম দা স্ক্রিন শেয়ারটা একটু করো তো বন্ধ করো তো স্ক্রিন শেয়ার চালু করো
Now I can do it again probably.
Now start
it again. Yeah, now it is fine.
Please don't do this. I'm requesting all the
please, please do not do that. Yeah.
Don't put your pointer on the screen.
Okay. See, again, someone is doing. Yeah, looks like. Anyways, so yeah, so let's look at that how solid phase peptide synthesis is done. So generally, people take it a resin. So I'm showing it here. This is the resin bead, which are made of mainly the polystyrene. Sometime nowadays, it's a big grafted resins are also available. Also, big resins are also available. But generally, they come as a bead. That's why the bead, one bead I'm showing here, and it displays the functional group. The chemist, what we do, generally, we modify with a linker. So why it is linker? Because after the synthesis of peptide, we want to clip that peptide out from the resin. So that is the way we can isolate that molecule. So this is the linker I'm showing. Let's say you want to synthesize IIT Ropar. So IIT RPR. So how it is being synthesized? First, we join one arginine molecule, and then further we couple with the proline Next, we do the arginine, and that is the way we do the chemical synthesis. So what we do generally, amine and acid coupling using a coupling reagent which dehydrate and form the amide bond. That's the way we join the amino acid and elongate the peptide length. So that is the way you can synthesize the IIT Roper. Now the question is, let's say you have here only 4 amino acid diversity in this particular. This is a just change here randomly position I to somewhere else, R to somewhere else, P to somewhere else. So how many peptides are going to be generated? There is a formula. So you'll be able to generate near about 4,000 peptide if you do the permutation and combination. But the challenge is, can you make the 4 peptides in a day, which would be crazy. It's very difficult to make this much of peptide in a day. But the technology, that's why acid kind of job. So how is it possible? That I'm going to explain now. Look at that, how do you make a library? Let's say as an example, this is called as a one bead, one compound combinatorial library preparation. So that means each bead is going to contain each single peptide. It's never going to mess up with the other kind of peptide. So one bead will be containing the only one sequence of the peptide, only one identity of the molecule. So now the thing is, let's, as an example I'm giving, the library of the pentapeptide, you are going to make, let's say, the having a 5 amino acid using a 10 variable amino acids. Amino acids, and while will be length of the peptide. Now look at, so 100,000 of it, still really a big number. How could you make it in a one day? It is possible to make it by the splits. This is the technology of the synthetic strategy, can be able to synthesize this much amount of peptides. Even not only that, you can make a 1 million of peptides. In this case, generally people use a particular type of resin, which is known as a tentagel resin, which is a pig grafted resin. The reason is that you can synthesize the peptide, as well as further you can utilize this particular bead for the screening, biological target screening. Either it could be cell, it could be the protein, it could be any other kind of target. That's the reason. So now let's take a look at an example. You want to synthesize a just tripeptide having 3 amino acids only, let's say. Only 3 amino acids, and the diversity of the amino acids are 3. DAV. I'm taking a very simple example so that you can understand. It won't make any complication. First, what you do, you take a 1 gram of resin. Let's say it's a huge amount of resin having a small bead of the resin, which is near about like 90 microns each side. Take a 1 gram of material and split them equally in a 3 different vessels. And after that, you synthesize the peptide. Okay, so now if you see the synthesize the peptide, one vessel will be alanine, another one valine, other one is aspartic acid. So now you have a only monomer in the resin attached. Now you mix them together. So now once you mix them and again distribute equally, then what is going to happen? Each vessel is containing AVD, AVD, AVD. All will be containing, all kind of resins will be containing of this kind of amino acids. Now, after that, if you again couple with the alanine with the one particular vessel, further the second one with the valine, and third one with the aspartic acid, what is going to happen? Particular this vessel is going to give you a alanine, 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 valine, alanine, aspartic acid. These are the 3 peptides you can make it. And here, if you couple it, it's going to have a AVVVDV. Here it's going to be ADVDDD. So that is the way now you again mix. So that is going to have a total of 9 peptide. Now 9 peptide you mix together and again you split. If you split, each vessel is going to containing again 99 peptide. And again, if you couple it, that is going to give you 3 to the power 3. That means 81 peptide. So that can be synthesized in 1 hour in my lab. So, but each and every bead, if you pick, it will have a only single identity of single molecule. So that is the advantage of that. Let's say what is going to happen after that. Now, basically, if you want to target any kind of target like a protein or cells or anything, you should have a process scaffold choice. Scaffold means it's a particularly, protein structure. Either it could be a random loop or it could be a helix, whatever I'm showing here. So these particular, you can take this kind of scaffold and modify or make the library with that. So there will be a big chance. If you know already target this, let's say what I'm showing here, targets are anthrax PA, anthrax protective antigen. This is the protein that actually delivers anthrax genetic material to the cell. That is the way it makes a pore and then do that. Ebola glycoprotein that I worked on in MIT, that is also a glycoprotein, is mainly responsible for the fusion process. And this is a thrombin. This is kind of a serine enzyme that actually responsible for the coagulation of the blood. And this is another protein is very well known, which is known as hemagglutinin. It's present in our cell also. Some virus also contains this particular protein. It's very interesting that this kind of protein that take a help and binds to our tract, respiratory upper tract and of the sialic acid specifically, and then they causes the viral infection. So we targeted this particular out of 4. We targeted this one, the hemagglutinin, this target, and we chose specifically this one, the random coil, because we know here it can fit only this kind of loop. Already it is well known that something interaction you probably know from. That's what I already showed, that you need to know first this interaction, need to know the nature, how nature does this. So if you know that is the way you can actually choose a scaffold. So in this case, we realize that this scaffold will be the ideal to target this particular hemagglutinin. So now how do you screen? That is the question. Now you make a library, let's say millions of members of library, each one has a bead and each has an identity of each peptide, individual identity. What you do after that, you take a protein, like it's a big molecule, and this big molecule tag with some fluorophore. Now together you mix in a buffer system. What is going to happen if particular sequence of peptide or particular, let's say, molecular integrity that matches with this particular pocket, that can actually bind this protein. And now you can look under the microscope. This is a real-time picture what I have taken. As you can see that not all beads are colored actually. Just few beads are colored. That means this particular protein which is having a fluorescence, that is binding to this particular bead. That bead displays a particular sequence of peptide. Now what you do, you pick all these beads, separate them and cleave the peptide out of this resin and then do the MS sequencing. The mass sequencing that will tell you the sequence of the peptide. And then further, you again synthesize this kind of peptide and validate outside by some biophysical assay, either isothermal calorimetry, fluorescent polarization, many other ways you can do it. And that is the way you can think about or develop the inhibitors. So this is the one just example I'm showing it here. This is the sequence that we found from this whole protein. This is here, it binds to the hemagglutinin. And we have done a lot of biophysical assay. We have purified this material and it binds very well to the hemagglutinin. Next, I'm coming to the fact that the technology what I have discussed here, the one bead one compound, it has a lot of actually pitfalls. Sometimes you may get a lot of false positive. They are promiscuous binder. They are very hydrophobic in nature. It binds to anywhere. So you may not be able to distinguish between the binders versus the promiscuous binder, which is non-binder basically. So you may find a hit. Also, you may not get a very high affinity binder through the OBOC. There is no guarantee. because whatever the peptides or molecules you are making, it's actually immobilized in the resin, the top of the resin. So sometimes it actually gives a false positive, or avidity effect it is called, because it has a many copies of peptide on the surface of the of a particular resin. So now, how do you mature the affinity? How you increase the affinity of a heat peptide? That is a big challenge. So it can be done if you let the molecules in the solution interact each other with a free thermodynamic moment, or having a proper flexibility or thermodynamic availability of their binding cavity or binding exposure to them. So this is called the solution in solution enrichment. Let's say you make the peptides, and these peptides you cleave in the solution and then incubate with a particular target and let them one particular binder binds. Further, what is going to happen? This particular complex is going to be very stable and other things will be around in the solution. Now you separate them out. So how do you separate them out? Now, this is one of the example I'm saying is here. Let's say you have a protein, which is an oncogenic protein, is known as the MDM2. Now you make a library, which is a kind of a very much targeted library. You cannot make here is one of the problem is here that you cannot make a millions of peptide library. You can have a very targeted library, a few numbers like 1,000, 5,000, 10,000, because solubility will be a big issue. That's the reason. Because it's not anymore in the resin. It's in a free solution. So now let it actually bind to the MDM2. And then further, what you do, you elute through the size exclusion chromatography using the buffer. Now you can see, this is the chromatogram. What I'm showing is, so the big particle, that means it's a protein which is bound with a particular peptide or a epitope that will elute first, and after that, with a long gap of the retention time, your small molecule, which is small peptide, is going to elute, and then you can easily separate them. And this particular elution of the bound particular epitope with the protein you can collect, and then further the interaction you can disrupt by using acidification, and then do the MS and get the sequence. So this is the way what we have done it. This is one of the very well-known inhibitor of the MDM2 for the cancer disruption or cancer inhibition, is well-known with the natural amino acid sequence having a KD, the affinity is 50 nanomolar, which is really is a druggable like. 50 nanomolar is a huge, great affinity, but still we wanted to achieve a higher affinity than that. So what we did, these are the actually three places what we have seen is mainly responsible for the interaction of the pocket. From this understanding, we had come through the crystal structure. Again, that's what I talked about the very initially. We need to know first the nature, how it does. So we realized that these are the three particular hydrophobic amino acids are the mainly the hotspot actually interacting with the MDM2. What we did, we take a 20 different varieties of the non-natural amino acid having a different functional diversities. As you can see, it's a 2-difluoro having just changed to tryptophan to something like, having a hexane just at the side chain, the cyclobutane instead of the, isobutyl group of the leucine. So these are the things we have done. These peptide we found to be like 100 times better affinity, which is 500 picomolar. This is 0.5 nanomolar. This is the affinity of this protein. That is the way we have developed. So you can see that you can have a heat particular by using a OC method, and further you can find a particular heat, and this heat you can modify, you can make a targeted library and go through the in solution enrichment to increase the affinity for this particular molecule. These are the first two steps if you want to generate the inhibitors. Then you can go for the biological process, pharmacokinetics, pharmacodynamics, toxicity studies, and many things you can do. But these are the first criteria, is that can you be able to do it or mature the affinity of the molecule as well as the selectivity of the molecule. So now we are switching the gear to the prospect in identifying the SARS CoV-2 hits. Using these technologies, can you do that? But before you go for, you need to know first the replication process of the SARS CoV-2, this particular structure of this virus. It contains like spike protein, envelope protein. I'm quite sure that you all of aware of right now. Its last 2-3 months is going crazy with the COVID-19. So you can see the 3 proteins I have here showing is a spike protein, envelope protein, membrane protein. These are the protein I'm showing. The reason is that you can look at these kind of protein you can target and have it. You can think about how you can generate a heat. So first, briefly, you can look at that what is the COVID replication process. Consider this is a virus, as I have shown in the last slide. Now, it actually goes inside the cell through the fusion process. A very well-known mechanism. The spike protein binds to the angiotensin converting enzyme to this particular receptor. It binds, and then further is actually cause the fusion process. The details I'll talk about in the next slide, how it happens. It's a very nice mechanism I'll talk about. And then further, it delivers the genetic material. It helps after that with the host ribosome. That means our cell ribosome. And then further, it translates to the replicase. It's the main protease, RNA-dependent RNA polymerase. This is the main enzyme or replicase, you can say. Along with there are some non-structural protein also translated, which are also known as an enzyme. They make a complex together. This again further, this particular, the virus, the genetic material, further it make a negative sense of the RNA genome. And this negative sense of the RNA genome further transcribed to the genomic RNA, whole the genomic RNA, or it can transcribe to basically nucleocapsid, single gRNA, or spike protein, membrane protein, envelope. These are the simple just single stranded RNA. It's transcribed. And from this actually further it get translated to the different kind of protein that needs to the viral assembly process or make the baby viruses that can go out and again cause the infection. But as you can take a look, this is the one here you can target. One thing that you can disrupt this virus to cell entry. Second thing, you can actually inhibit the replicase. This is another process you can think about, that if you inhibit this particular replicase process to make a complex as well as the particular transcription process, that way you can stop the replication process. And fourth is what I'm considering, that viral assembly, which generally people do not consider this particular process, but I think this could be one of the important also. That let's finally how it assemble. And during this process, can you inhibit this assembly process? That is the way, these are the 4 steps you look, and you can try to inhibit or generate an inhibitor using the technology what I have described. So how could you do that? First thing, you need to know how the fusion process goes. As I take a look, this is the virus and this is the spike protein. This membrane is for virus and this membrane is for the cell, normal human cell, think about. So what I'm showing here is an angiotensin converting enzyme to receptor it is. The spike protein has a S1 and S2. It's a sub-particularly type, and this particular envelope of the S1, it binds to the receptor and further it gets cleaved off by the enzyme. And automatically it exposes the S2, which is the spike protein, is a hydrophobic triple helix bundle which binds to the outer membrane by the hydrophobic interaction. And then what happens, you can take a look, the next process, it basically pulls our membrane to fuse with their membrane. And that is the way the driving main force is basically the hydrophobic interaction between the blue color as well as the green color. So that is the way the hydrophobic interaction as well as some ionic interaction, they pull together and make a 6 helix bundle. As you can see, the 6 HB means 6 helix bundle. So here is the fusion. So both now the membrane is the same. Now there is you cannot distinguish between our cell membrane versus the viral cell membrane. That is the way once it fusion done, then the virus delivered the genetic material to our cell, and then the replication process goes. So how could you stop this particular fusion process? So you can do one thing, think about, so let's say this is the infection it can have by the interaction. You can think about that you can block the ACE2, this particular enzyme receptor, so that it cannot interact. So you can think of either like, as antibody or any kind of receptor or kind of small molecule you can think of, so that can actually block this particular ACE2 angiotensin converting enzyme that can block. So that is the way it cannot interact the coronavirus. And the next would be like, if you can think of generating an antibody, or here is the another thing that you can think of antibody mimetic that can bind to the S1, spike protein S1, so that it cannot interact with the ACE2. So we need to know first which particular protein portion interacts with the ACE2 receptors. So recently there are a lot of papers, that's why it appeared in Nature, Science, and many other good journals, that already people have deposited the recent COVID-19 related their protein structures or interaction with the spike protein and angiotensin converting enzymes. These are all structure has been deposited, and these will be really, really helpful for the further drug design purposes. So that is the way you can think about this. Is another process you can inhibit the virus to interact with the ACE2 enzyme. So next I'm coming to this is the 3 process I have talked about, and this is another thing you can think about, that what I talked about the ACE2, particular the bundle, helix bundle formation, 6 helix bundle formation. So that driving force, the hydrophobic interaction between this green and this blue, that you can disrupt. Think about this is the whole helix, 3 helix structure. And then if you have another complementary helix that can sit on, so that they cannot bend, they cannot pull. So the hydrophobic interaction is already satisfied by the foreign substance, which is a therapeutics, peptide therapeutics, so that there will be no more interaction. So that is the way you can inhibit. So this way, there is one of the very well-known drug has been discovered for HIV fusion. It's known as a T20. You can search in the internet and take a look. Okay, so this is the one of the another way you can inhibit the fusion process. Next, we are going to the RNA-dependent RNA polymerase, which is a 3C-like main protease inhibition that we talked about, the replicase, the RNA-dependent RNA polymerase. So see, look at this structure. It's already deposited in the Nature. This is the red color, the circle, what I'm showing is, this is the enzyme cavity or pocket, you can say. The better to tell as an enzyme, the active site. This active site is mainly responsible for this particular replication process, which is having the transcription process. And here, if you block it, this particular, the pocket of this enzyme, that active site, then you can be able to disrupt the replication process. So this is already a well-known, and again, it published in the Nature. You can see there is a peptidomimetic. It's a small peptidomimetic has been discovered, which is IC50 is 1 micromolar. It's a very good druggable candidate. And this fits into the active site of this enzyme very well. And I'm showing here that how they have already published how this particular peptidomimetics occupy the active site of this particular enzyme. Interestingly, it is a cysteine, it contains a cysteine in the active site. And cysteine, you know, it's a good nucleophile. And they have designed a covalent inhibitor. As you can see, this is a Michael acceptor, which is having a alpha beta unsaturated ester. And this is a double bond, and is cysteine sulfur is minus sulfhydryl. It can attack over this particular carbon, which is the beta carbon of this particular amino acid. And that is the, here you can see the connection between the cysteine and this molecule. And after that, this molecule, the enzyme basically modified with the covalent bond. It's not anymore only the non-covalent, it's all as well as the covalent bond. So till this, you know, after targeting this kind of peptidomimetic to this RNA-dependent RNA polymerase, till this enzyme get degraded into the cell, this enzyme is inactive. So there is a no more replication is going to happen. That is the way you can inhibit the process. So next we are going to the possibility that I talked about. I consider this is one of the, this can be considered also. So my research group is also interested to do that. So disrupting the virion assembly by inhibition of the protein-protein interaction between the envelope protein and the membrane protein. So during the virion assembly, what it does. So once, you know, I have shown it how it replicates it goes through the process, and then further it makes all the protein it needs to again make this virion or the virus, baby virus. So what it does, as you can see, this is the one of the structure, the way it is assembling. It's a zoomed structure. As you can see that in the cytoplasm, what happens, all the proteins required to make this virion assembly, it's already together. Now it needs to cut out, makes a bulb first, and then further make a virus. How? It interacts the E and M protein. The envelope protein and the membrane protein, they interact, as I'm showing with the red color as well as the green color protein. So it's very well known, this is the E protein structure. It's a very small protein. The C terminus, it interacts with the M protein, and that is the way it gets seasoned by their own interaction process, and the viral assembly actually happens, and it buds from our cell. And there are many of virus, babies viruses get translated through the process, and then replication happens, and then further it buds from our cells. That is the way it happens. So now what you can do about this? So if you can design a peptide, let's say either you can take assistance of this technology, you can screen this particular C terminus of the E protein and find a specific peptide binder that can block the C terminus of the E protein. That is the way you can block this interaction between the E and M. And therefore the virus assembly get disturbed. And already it is well known previously different biological studies that without this interaction, virion assembly is possible, but it may not contain the E protein. The E protein comes out from the virion assembly, and if any virus doesn't contain this E protein, then they attenuate their activity. So their potential for the further infection goes down. That's called attenuation. It attenuates their activity. So that is the way, that is why I am considering the E protein could be a good target. So we can think about this. So this is only possible, like, these are the targets. It's a already no one has explored all these things. How could you develop a peptide binders which can selectively bind this and inhibit the viral replication? The only way you can think about the de novo screening. So that's what the technology, what I have shown, that OBOC 1B1 compound, you can do a high throughput screening and find a further.
20 participants I am watching here. So
Hello.
Participated through Zoom. So today I feel honored to be part of this.
So basically what happens after that, if you have a hit, then you can go for the affinity maturation that I showed you, the in solution approach. So that is the way you can develop the inhibitors. So what with this, I would like to conclude that what I have discussed, that this first thing what you can do, you can inhibit or develop inhibitors, the fusion peptide, basically that you can have a S2 either or a S1 epitope that can disrupt the receptor S1 interaction or the fusion process of the S2. The second one, what I have shown is RNA-dependent RNA polymerase, the active site of this particular enzyme, you can block by a particular peptide, shown as an example of peptide mimetics. It's published recently in the Nature with the crystal structure. And also the third thing is the assembly, which is people have not considered yet. What I think would be a good another strategy, that you can block the C terminus of the envelope protein of this, the SARS CoV-2, and that is the way you can stop the Cesium process or viral assembly process. Or if even though virus can go out, the assemble, lack of the E protein can attenuate the activity of the infection. So that is the way you can inhibit the this particular COVID-19. So this is only possible, the technology that kind of I have explained, the OBOC and in solution enrichment technology can be helpful. That's what I think, and you can find the hits out of it. Further, you can go for the other biological studies to understand it can be applied for the drugs purposes or not. So with that, so my lab is basically working on the biomimetic chemistry, high throughput screening for the recognition of disease-related biomolecules, and finding biomolecules and trying to apply the further discovering the diagnostics as well as developing therapeutics, and also working on the bioconjugation purposes. So mainly what I'm working with the molecules are the engineered peptides and mini proteins, that is a small protein. So I think about the atomic cutoff balance, which can have a better, the cost-effective therapeutics, I can say. So that is the way also you can handle any kind of small molecule. There's structure diversity you can create, and chemically you can modify them indifferently, and that is the way you can generate a easy available molecule. So my target is mostly like in a cancer biomarkers, different kind of glycans, lipids, as well as the protein-protein interaction, that what I have shown. Also, I'm working on the viral proteins, the fusion proteins for the controlling or combating of the tropical diseases. So this is the IIT Ropar, the dynamic and vibrant, very nice, the campus, the night side of the look. This is the gate, basically. We have a very nice gate having a, ancient kind of Harpan's civilization with the sculptures and with the, Cuban, or the steel Cuban having a chemistry top of it. And this is the top picture from all the buildings, quarters, hostels. And this is one of the hostel building. And this is one of the departmental structure. So our department structures are looking like that. And recently we are holding the second rank in India Times with their ranking. And with that, I would like to acknowledge my collaborators from the MIT, as well as my PhD students, Mr. Saurabh Chatterjee, and last, the summer intern, the Nitesh Mani, and also as well as I would like to thank the DARPA Novo Nordisk and for their help in funding also the IIT Ropar, definitely. And with that, I would like to thank, and if you have question, I can answer. And thank you for listening.
Thank you, sir, for the wonderful discussion. Now the session is open for questions. We already have a couple of questions with us. So I'll ask on the participants' behalf to Professor Bandyopadhyay. the first question which has come to us is, sir, what is your opinion about this virus? Is it man-made or natural?
Okay.
Can anyone make virus like this in lab?
Yeah, I mean, just I would say this is very much like a debating right now. And also, I would say that this kind of assembly of this virus is possible. First of all, this I would say, because the Wuhan, the lab, the virology lab, they do have, and they have published many papers, specifically one particular PI who has a lab who has been assembling last 5 to 6 years the different kind of viruses. They can mutate, they can do many kind of things. So that has been actually already they have reported in a very big journals. And there are big questions regarding that. Already people have commented from the different countries over like US as well as the France, that what is the use of this particular viruses you are assembling. So anyways, but kind of people are thinking right now it's kind of a leak from this particular lab. Maybe somehow it's contaminated due to some negligency or safety violence. Somewhat people are thinking right now, but I do not know that exactly until unless things are revealed. Okay, so the newses are coming like that. But it is possible. The first answer is the scientific answer, that it is possible to assemble this kind of virus because there are many good journals. There are Nature's as well as the Science that particular lady PI has already published.
Thank you, sir. So next question we have is from Sumit Kumar Panja. His question is, how do we detect such virus having trace amount in human? Nice presentation. I think he's asking about some kind of diagnostic tools or something, I guess.
Yes, yes.
How do we detect such? Yes.
Yes, so basically that is one of the big problem right now, that the amount limits actually the diagnostics problem. So that is the reason sometimes you need to do many times their genetic material extraction and other thing. That's why it is possible. So what happens, you can get a small amount of virus, get the genetic materials out, and further you can do the PCR. Okay, so this is a kind of a reaction you can do in the help of enzyme. The PCR, that's why it's telling the people are doing the RT-PCR and other thing. That's what keep showing into the different medias as well as the TV, other thing. So RT-PCR means it's a PCR, polymerase chain reaction, that you can take a help of an enzyme and you can make 1,000 of copies. Millions of copies. That is the way you can increase the amount and further confirm this genetic material corresponds to that particular virus or not. That is the way you can confirm.
Okay. So thank you, sir. The next question is from one of our faculty members from the Department of Chemistry, Dr. Namrata Rai. She has asked, she has firstly complimented, excellent talk, privileged to listen to you, Professor Banerjee. I would like to ask you, to what extent is antibody-antigen interaction affected upon immobilization of either one of them?
But okay, this is a good question. Yeah, so I think this is all about the affinity again. So the affinity is, again, the fact is, like I talked about, the affinity is how tightly it binds. So now if, let's say, antibody and any antigen has a picomolar or nanomolar, a single digit nanomolar affinity, then you will be able to definitely trace a particular concentration. Of course, even lower than that. But if you raise any antibody which is having a weak affinity, it will be very difficult, even though if you have antibody. Okay, so that's the reason it is kind of a question that you need to know first the affinity of this antigen and antibody. So if it is very high affinity, of course, you will be able to get it in an automolar concentration even. Like even if it has a very, very high affinity in a picomolar between antigen and antibody. Is it, I answered the question?
I guess so.
Yes, sir. So thank you. Thank you. I was the one who was asking the question. Thanks a lot.
Yeah, okay.
Sir, what I would just like to, comment on is that if the antibody is immobilized on a surface, it
Right.
needs to be in a manner that it's that Fab region is not affected, right?
True.
I mean,
True.
is there any way that we can actually control that? Because when we are
Yes.
immobilizing it,
Yes, you
because
can do
if that region remains unaffected, then I think the binding should not be affected as well, right?
Yes. Yes. 100% you can control it. So how could you do
Right.
that? I can tell you. This is again, it's a bioconjugation. Okay. So
Okay.
let's say that if you look at the antibody, the Y-shaped I showed, if you look at the C terminus, the Y, okay, like the Y I'm showing here, can you see my screen?
Yes, sir.
This
Yes.
is Y. So the bottom Y is known as a C terminus of the antibody. Okay.
Right. Right.
Now this place, if when you actually express any antibody, this place you can put a tag, a peptide tag. Okay. So that
Right.
peptide tag could be very specific for immobilization with the chemical modification. Like let's say if you end up with a cysteine, let's say, or something, you can keep it as a free cysteine, or kind of the small affinity tag you can make it. So that can be immobilized to a particular surface. Possible and
Right. Right. Okay. Thank you so much, sir.
Yeah. Thank you.
Okay, thank you, sir. The next question is from Shayok. He has asked, infurfatide is recognized as fusion inhibitor. Does it have any application in COVID-19?
Well, so this is, so this is basically what I talked about, the T20. Okay, so this is the HIV fusion inhibitor. So this sequence, you have to look that this particular sequence, the T20, that the drugs you talked about, it doesn't actually stop the fusion process, the 6 helix bundle, the driving force that needs to make a 6 helix bundle. It's not going to stop because it's not a complementary sequence, probably. Because the S2, if you look at the sequence doesn't match with the HIV properly. So it has to be very specific, as well as the affinity should be very high. Then only it can disrupt this kind of pulling the assembly of the triple helix to the 6 helix bundle structure. Is it okay? Yeah. So it has to be very specific. Then only you can. Probably it may not work. What I think, because sequence are not same.
Okay.
Yeah, Prabhupada, I have a question.
The next, yes, sir.
Yeah.
Yeah.
Thank you, Anupam, for your nice talk.
Yeah,
but
thank you so much.
I just want to know, I want to know just, people are trying for vaccines, right? And we do not know when it will be coming. But vaccines are based on several things, like, it's could be virus-based, could be viral vector-based, could be nucleic acid-based, RNA and DNA-based, and could be also protein-based vaccines.
Protein and peptides also.
Right, peptide also. So
yes.
My question is, many researchers, they are trying to inject directly the coronavirus proteins directly into the body. That is one way of looking at things. But few people are also trying fragments of proteins, or protein cells that mimic the coronavirus outer coat are also used.
The fragments,
so
yes, the epitopes,
your...
particularly, yes. Yeah. This is the one of the approach. I think this is really develop the peptide vaccines this way, and this is very powerful if it is going to work. If it does work, that's very, very powerful actually.
Yeah, so that's all. I think this will be, apart from the nucleic acid based, or the directly viral vector based, this protein based vaccine will be more useful, I guess.
True. I completely agree with you. So I am working on this this direction already with we have another professor here, Professor Agrawala, Jain Agrawala. So so his approach is kind of like that. So we try to find out epitopes. If let's say the what I have shown, the S1 protein, the spike protein, the S1, that particular any of the peptide fragments, which is known,
that mimic the coat.
Yes, so which is known not to mutate too much. First, we need to know that if it mutates, then it's not going to work actually. So if it's supposed to not mutate as well as is exposed to the plasma. That's another thing, because antibody, then only antibody is going to recognize that. So they find out a lot of this kind of peptide epitopes from this protein, from the S1 spike. And then what we do, we generally try to just screen that, that's all, that how it is working. Can we raise any antibody against it? And that antibody can neutralize these viruses. That's how we generally go.
Okay.
Very powerful approach, and thank you for raising this.
Yeah, many companies are trying for this particular approach, I guess.
Right. I agree with you, yeah.
Okay.
Okay, thank you, sir. Okay, next question is from Tubai Chaudhary. He has asked, sir, is there any synthesized peptide mimicking ACE2 receptor?
Okay, synthesized peptide that can mimic, yes, it is possible. Of course, this is again, you can call them as a kind of ACE2, the receptor mimicking peptide. So far it's not there probably, but people are trying, to develop this. It's possible, like if you can develop or mimic the particular receptor mimic by the peptide of ACE2, then it can bind to the coronavirus. So that is the way you can stop the interaction between ACE2 and the coronavirus and inhibit the infection.
Okay.
Hi, Professor Anupam. This is Ravi Kumar from Portugal. So I'm alumni of ISER Pune. So I have one question. Is there any way of detecting COVID-19 by using chemical-based method other than PCR method?
Oh,
so
yes,
like
of
a
course.
fluorimetry or colorimetry method?
Well, so yeah, I think there are many things people are trying to do that. So let's say as an example, if you have any specific, again, either a small molecule or peptide that can bind to this coronavirus, it is definitely possible to do that. But I don't think so far something has been already come out in the market. People are trying in a different company. People are actually trying that. The way what you say, people are trying actually. I'm getting some information from different companies that people are trying that. But so far, I don't think any 100% success is there yet. But people are from the Delhi, IIT Delhi, people tried to mimic some of the epitope of any antibody that can bind to the spike protein. That is the way you can achieve the diagnostics also. So that people,
many people are actually trying for easy method, not PCR, but something like in a spectrophotometer or fluorimeter to have a tag, something like which fluoresces something. People are trying that. I've seen some reprint of these papers, but those have not been established.
Yeah, it's not like in the market yet.
No, not yet.
That's right. Yeah.
Thank you so much.
Yeah.
Okay. We have a few questions on YouTube as well. I'll read out a few.
Yeah, sure.
Amit Maiti, he has asked, does sanitizer or 70% alcohol really kill the COVID-19 viruses? Amit Maiti.
So yes, so this is a good question actually. So again, this is kind of a, so what happens that in presence of a 70% isopropyl alcohol specifically I'm talking about, if you need a ethanol, you need to have a 80%. Okay, so that's the criteria of the WHO mandated, how we make the sanitizer. So this liquid sanitizer, if you have a 70% isopropanol with a water, that can kill in terms of, it disrupts the membrane, viral membrane. So viral has to make it has a membrane actually, a bilayer membrane bilayer. So that contains the all protein, what I have shown the picture, so that's the thing. So it disrupts the membrane, as well as sometimes the proteins are there, which is holding by this membrane. It gets also, their structure gets disturbed, is denatured. So slowly what happens, the structure I told initially that you need to have a proper folded tertiary structure to have protein as a functional material. So that if get disturbed, so automatically what happens, you can understand. So it actually lose their activity. That's what it happens.
Thank you. I hope the question is answered. We have a few more questions here. Just a minute. Yeah. Can peptide mimic alter cytokinin interaction, which is inhibited by COVID-19? Indranil Chatterjee. Question from
Oh
Indranil
yes,
Chatterjee.
Of course it can. So basically what happens, see, if peptide can interact with or peptide mimic can interact with any kind of protein. Okay, the cytokine is also, again, it's another kind of a protein. Okay, so if it can bind a particular, it has a cavity, or even though if it has a surface also. So that's the beauty with the peptide is peptide can target the surface as well as the cavity, both, or pocket, let's say, or enzyme active pockets anywhere. It can do that. But any small molecular size, if you look at, it can only find the pocket. If you need to interact with the surface, like a big protein interaction surface, the limitation of the small molecule is there only. So it cannot do that. But as peptide has a huge structure, and it takes a kind of, their own secondary different kind of structures. So it can give a big, huge surface area to interact with or inhibit with a protein interaction. So of course, it is cytokine is possible. Of course, it can possible.
Thank you, Professor Bandyopadhyay. We have a few more questions. This is from Saurabh Banerjee. Sir, by using PCR and gel electrophoresis and detecting active genetic part, if we use genetic engineering to cut it out and trying to make a vaccine, is it possible?
So, okay, I think this is mostly like a genetic engineering question. Okay, so this is very much like a technical question. I may not be very suitable person to answer that. Sorry about this. So I would say, like, you can talk to some genetic engineering person. That could be much better. He could give you better, or she could give you better answer.
Yeah, thank you, Professor Bandyopadhyay. Now, this is another question. How can we know the shape of a peptide 2 to 3 nanometer if anyone mimic the structure and receptor as ACE2?
Can you read it one more time?
Yes. How we can know the shape of peptide 2 to 3 nanometers if anyone mimic the structure and receptor as ACE2?
We know the structure. I think you have to crystallize them, or maybe what you can do, see, you can basically draw the structure in any ChemDraw. There is a one software, you know that probably if you are a chemist, you know that ChemDraw is a structure. Okay, you can make a ChemDraw as a structure and then go to the 3D formation. You can make it a 3D structure out of it, and there are tools that you can get the size of this particular any molecule. So that is the way you can understand the what is the particular length of the peptide, or does this mimic the CO2. You can easily understand by that way.
Sir, actually I am asking that
crystal structure.
Hello.
Yeah.
Sir, if anyone mimic the ACE2 receptor for binding that SARS CoV-2, and after we check the ELISA technique that it is really binding or not, and after that, if we substrate at one graphene substrate to attaching with a linker like PBS and anything, and make a device, and that for that reason, if I go for 10, can I recognize that this is my shape before the attaching the virus and after attaching the virus? Can we got different image?
I didn't get your exact question actually. Can you one more time repeat, please?
One ACE2 receptor mimic peptide is synthesized, and it is attaching the virus, and we got the from ELISA we can confirm the peptide are attaching the virus, and when we make a device like base with graphene or graphene oxide,
Okay.
and go for 10 images, can we check the the receptor is attaching the virus or not? Is there any shape changes in 10 images? Can we recognize?
Yes, of course. TEM can do that. TEM can do it in a nanometer range, right? If you have a high, like a high resolution TEM.
You can face correlation in HRTM in face correlation image. You can.
Yes, yes. If you can have a high resolution TEM, definitely you would be able to get it, of course. But these live viruses, playing with these live viruses is very dangerous. Be careful with that. You need to have a proper lab. Okay.
You can do it Sargoroti virus.
Yes, you can take it as a proof of concept. If you want to have an idea, this you want to basically have a proof of concept. You want to have the data. You can take it a very different kind of viruses which are not infectious. You know that, and then try this particular module first as a proof of concept, and then after that you can go for.
Actually, sir, the peptides are basically carbons, and then graphene is also carbon. And my concern is that there is very dilute concentration contrast difference, and how can I determine?
Right. So basically, what happens? I can tell you. It's again the question asked that affinity is the matter, of course.
Yes.
Okay. So affinity, if you have a very high affinity peptide, it is definitely going to recognize. But another point is that affinity, what we are talking about is mostly like a one-to-one interaction, right? Now, once you immobilize any peptide to the surface of the graphene, that is going to make a multivalent structure, the multivalent. That means it is kind of, it's called avidity effect. Okay. It's a multiple copies are there. So automatically what happens, the affinity increases by the affinity effect. Okay. So that is the way that you can definitely be able to recognize, but you make sure that you have to have a really high affinity binder, peptide binder. Otherwise you may not be able to, capture that.
Yeah,
that's the one.
We have to check at first ELISA or any other things that it really bind the, and how below level, what is the detection limit.
Yes, the limit of detection you have to go for.
And we have to check that also the swab collection, CMT virus also it can detect or not. Then we can only the bind with graphene and then.
Sure.
And we can do it better in CD also, circular dichromism. We can do better experience out of TEM.
So basically let's say if you have CD will be a little bit complicated, I think so. The reason is, let's say if you see any peptide, let's say it's a just linear peptide. Now, this linear peptide, if it binds to the coronavirus and then it changes the structure, that is the way you are thinking maybe going to predict by the CD, right?
Yes.
But the coronavirus contains a lot of protein that will interfere.
Yes.
What is the change is going to happen? You may not be able to understand what I think.
That's the main problem. How can I recognize that? And and graphene also good SPR.
Yes.
If the peptide also good SPR, from Raman you can also got, but there is also the contrast problem.
Well, I think you can go for the ELISA. You can take a help of the secondary antibody, right?
Yes.
So like you have a epitope immobilized to the ELISA plate, and then you put the virus, then you drop cast the fluorophore labeled secondary antibody. That will tell you.
Okay.
Isn't it?
Yes, sir.
Yeah. Okay.
Yeah, since we are running short of time, we'll just have one or two more questions and we'll end this question answer session. So we have a question from Mohammad Maqsood Ahmed. His question is there any disadvantage of split pool synthesis of OBOC combinatorial library preparation?
Yes, what I mentioned already. Yes, I already mentioned that. What is the problem? The number first problem is, let's say you want to make a huge longer chain of a peptide. Okay, that is going to be a problem. I think so, because the reason is, once you synthesize a peptide, after 15 amino acid to 20 amino acid, it gets aggregated. So what is going to happen? There will be a problem in terms of the purity of the peptide. That is the first. Second thing is, talked about the promiscuous binder. That means it's a like a this kind of sequence of the peptide. It binds everywhere. It doesn't matter. It binds everywhere, so that you will be end up with a false positive. That maybe you see as a this is a binder. You can think, but when you synthesize and validate the biophysical assay, you realize that that is not binding at all. That means this is just a false positive. That's the second thing. Okay. And the third thing is, as I talked about just right now, the avidity effect, the multivalency. One particular bead contains lot of copy of the peptide. So what is going to happen? You may not be able to get a high affinity binder. So as it has a multiple copy. So due to avidity effect, even though if it is a low micromolar binder or moderate binder, you can say, it can bind tightly. Even though if you give a wash, stringent wash, it may not wash out. So that you will be end up with that particular peptide. You think that this is a good binder, but it is not. So the three things I talked about, these are the major problems. But the affinity, you can definitely, affinity and specificity, you can cut off further. If you have a hit, then go for the in solution enrichment, the solution targeted library screening process. I hope I have answered.
Thank you, sir. So last question for today, and this kind of sums up everything. So I'll just read it out and put it in this way. This question is from Pranjal Pine, and he has put his question on YouTube, and he's from, he has introduced himself from the Department of Chemistry, Vishwabharati, I think, yes. If such therapeutic drug is discovered somehow, what are the possible steps to see drug is effective before applying to human body? And if any fruitful results arrive, how much time it will take? I think the last part of the question is something that we'd all like to know. How long can we wait? Like, is there any
this is, yeah, of course, it's a really good question. So to answer this, I would say first thing that, how much you are putting effort, that matters the time. So let's say if we are behind COVID right now, the way the diagnostics and other thing, vaccines, these are the things. In a month of time approaching and people are trying to discover these things, probably you might have not seen before. The thing is now the reason is we are putting a lot of effort in this particular research. So therefore it is progressing very well. So that's the one thing, that it depends on the how much effort someone is putting. And another thing is, let's say if you have a particular inhibitor or binders, you can say, and then to come to the human, it takes really like a long time, because after we go for the PK/PD studies, which is called a pharmacokinetics and pharmacodynamic studies, the distribution of the drug, and that the long time it is actually incubating in our body, how much renal clearance it's going, how quickly out. If it is going quickly out, then we have to think about how to retain this drug into the body. So you need to have a modification, otherwise you cannot target that where you would like to target. So these are the things. And third thing is like toxicity is the another matter, the how toxic it is. All these things will tell you it takes some time. So people try in first in mice, and many mice you have to take it. These mice further, some mice may respond very well, sometime some group of the mice may not respond in the next batch. So a lot of things, actually there are caveats. Okay, so everything may not go very smoothly. So that are the hurdles are there always. So that is the way it always takes long time to do all these things, until unless we get a very good information that okay, things are really great and have a green flag to go to the human, we can do it. So it takes long time and many other factors, a lot of things matters actually. So it's quite difficult that what would be the timeline or anything. So as an example, as a small molecule discoveries, like generally it takes sometime 5 years, sometimes it takes 10 years also. Okay, any therapeutics or like a protein therapeutics, peptide therapeutics are mostly coming a little bit in a lot of pipelines and clinical trials. The reason is they are more biocompatible, bioavailability is more, and they are they randomly do not interact with the cell or enter into the cell. The small molecules go here and there into the cell, and then it's very tough to control them in a in a particular manner. So a lot of things matters. That's why you can see in a pipeline of the clinical trials, a lot of peptides and proteins are now in a days, and FDA is approving a lot of therapeutic peptides as well as a protein rather than small molecules. That's why small molecules are taking long time, and now companies are not much interested to invest a lot of money to the small molecule drug development. Even you can see that a lot of reviews are coming right now. So because the reason is there are a lot of data bank is already, they have drug banks, and they are trying to repurposing these drugs that they already known to their toxicity level, other kind of testing PK PD, and they can be utilized further for some other disease purposes. That's what they are calling as an orphan or the repurposing of the drug.
Yeah. Thank you, sir. So with that, we come to the end of the question and answer session. On behalf of the organizers and all the participants, I sincerely thank Professor Bandyopadhyay for this excellent talk, which is very timely. And thank you for patiently answering all the questions from all our participants. Thank you, sir, again. May I now request Head of the Department, Chemistry, Dr. Arpan Manna, to kindly give the closing remarks. Dr. Manna.
Yes. Am I audible?
Yeah.
Yes, yes.
At first, I am giving a big thank you to our speaker, Dr. Anupam Bandyopadhyay, for his timely and his efforts towards delivering a timely speech, actually, which is really interesting, because I have already told earlier him that most of our listeners or the participants are maximum from the graduate or undergraduate or postgraduate students. So he also told me that I will start from the ABC of this technology assisted antibody mimetic discovery. And that's what he had done. And it was really overwhelming. Actually, I am from physical chemistry, and I don't know very much about this protein and peptide related chemistry. But through this talk, it is quite lucid to me. I hope all of the participants have really enjoyed the talk, right? So I would like to express our profound gratitude on behalf of the Department of Chemistry of Adamas University. Thank you, Anupam sir, for agreeing to deliver such a wonderful talk to all of us in such situation. And also, we know that there are technical glitches last time, but still you all yourself have kept patient, and also again you have told me that you will again deliver the talk at whenever we shall decide the time. So with this, I am also requesting all the participants, basically the registered participants, that shortly we will send you one feedback form, right? So we know that we had almost 465 registered participants. So as soon as you fill the feedback form properly with your name as well as your institution, then you will get the participation certificate. But due to the limitation, it may take 6 more days from today, actually. But I would like to request all of the participants that please cooperate with us and fill the feedback form that will be sent to your registered email ID, where you have through which you have registered the Google form earlier. Also, I would like to express our profound thanks to our Professor Sanjeev Ghosh sir for being all-time an enthusiastic person. And also I would like to thank our Dean sir for inspire us for holding such type of wonderful webinar at this pandemic situation. And also I would like to thank all the participants, those who have asked question, who have not asked question, and through both the platform, that Zoom and YouTube, those who have viewed this talk, for keeping their patience. So there are some technical problem, but hope with the help of our speaker, we could have avoided that technical problem. So I again want to thank you, all of you. And also I want to thank our Chancellor sir, and for providing us such a platform or opportunity to hold such type of wonderful webinar. And I thank to all my colleagues, especially Dr. Prabuddha Bhattacharya and Dr. Deepsika Bhattacharya, who take their pain to host this seminar. And Dr. Prabuddha Bhattacharya is a renowned organic chemist, and Dr. Deepsika Bhattacharya is a renowned nanotechnologist or nanochemist actually. And also I want to thank all other my colleagues from the Department of Chemistry and all the faculties from the School of Basic and Applied Sciences who had extended their great support to hold this webinar successfully. So I would like to express vote of thanks again to our honorable speaker. Thank you very much, Anupam sir. I want to tell another thing that Anupam sir is an excellent vocal singer of Rabindra Sangeet actually, because I first met when in Pune, Riser Pune, in the hostel. So during my NCL Pune time, we hold a joint Saraswati Puja at NCL clubhouse. So there he was invited all the time for singing Rabindra Sangeet, and he was an excellent singer. Thank you, Anupam again for
Thank you so
joining
much,
us.
Arpan. Thank you. Thank you for the organizer again. Thanks so much. It's my really pleasure to give a nice present my research talk here.
So thank you, Dr. Manna, for your kind words. On this remark, on behalf of the Department of Chemistry, School of Science, Adams University, I thank everyone, every participant. I thank our speaker, Professor Bandyopadhyay, for his time, Professor Sanjeev Ghosh, Professor Bimal Kumar Sarkar, our honorable dean. Thank you, everyone. So stay safe, and hopefully, with the amount of effort that the scientist community is putting up all around the world, and especially with efforts of kinds like Professor Bandyopadhyay, we'll be soon getting a vaccine or some kind of therapeutic treatment for this deadly virus. So till then, stay safe. Thank you all, and we end this webinar.
Thank you. Thank you so much. So, Arpan, I'm leaving.
Yeah. Okay. Thank you very much.
Okay. Thank you. Thank you. Thank you.
So, Arpan, may I leave?
Yes, sir. Sir, if you want to tell something, you can tell.
No, it's, they were not completed. Concluded.
Thank you, sir, for joining us.
Thank you.